Great. Well, it's a great pleasure for me to, oh, I guess first we should mention the sponsors. So we have a sponsor, Nexomics. They sponsored our beer and pizza today. So if you guys ever encounter them, you can say thanks. But besides that, it's my great pleasure to introduce Yunha. Yunha has been working with me on lots of cool projects in the past, and I believe we're still collaborating. And she comes all the way from Harvard, which is, I think, at least 20 minutes from here. And she recently got her PhD. So it's Dr. Yunha, I guess, now. Yeah. You take it off. Oh, and I forgot to mention, she also has a cool new nonprofit company, and also potentially recruiting people. So if you're interested in whichever she's presenting, come afterwards and talk to her, because I think there might be some cool opportunities there.
Well, thanks, Sergey. All right.
Oh, I should just make sure that it's sharing on Zoom.
I think it is. Okay. That means I think it is. Okay, cool. I hope so. All right. All right. Hello, everyone. My name is Yunha, and I'm really excited to be here. So the work I'm going to be presenting today started at Harvard, and now it's sort of gotten into this sort of like nonprofit scientific research project of genomic language modeling and building sort of genomic intelligence to understand genomes better. So the problem that we're trying to tackle in biology, and I think one of the biggest problems that we have in biology is that there is very little functional information when you look at the annotations. It's estimated that less than 1% of known gene sequences have a validated function. And we probably only scratched the surface of sequence diversity. In this new paper by folks at JGI, they discovered that as more samples become available, protein sequence cluster number increases linearly for reference genomes, but exponentially, and without reaching a plateau for metagenomes. So metagenomes are genomic data that come from environments. So you literally get a piece of soil, and then you extract DNA directly from the sample without the need for culturing. And that way you can really get the full diversity of genomic information sequenced and on your computers. So because the label information is so scarce, we need an unsupervised and high throughput method to accelerate sequence to function prediction. So in this talk, I'm going to talk a little bit about how structure prediction is different from function prediction, or function prediction is different from structure prediction. And I'm going to talk about the genomic language modeling work that we did at Harvard in collaboration with people in academia. And I'm going to then talk about the work that we're currently doing on improving genomic language modeling, and talk about sort of the consequences of this work. Okay, so structure is really important, and we have made a great progress in predicting structure, but it's really far from everything we need to really understand biology. Function cannot be predicted directly from structure. Even when you have a structure that looks really cool, it's difficult to tell what it does by just looking at the structure. Proteins also do not exist in a vacuum. Other proteins and environmental conditions govern their function. And proteins with similar structures can perform different tasks. Yeah, I don't know how to turn that off.
I think we have to turn off.
That's complete.
Actually, no, I think what we need to do is we need to go down to Zoom. Where's Zoom here? Oops.
Oops.
Sorry about that. How do we get back to... Hopefully there's nothing too sensitive there. So actually, I think if you make your full screen. Oh, here we go. Yes, yes. It might be manage participants. Yep. And then click more. Hit play, join, leave sound. Yep, that's it.
Okay, now
Yeah,
it should be
so now, yeah, but
Okay,
people
cool.
show up there. I mean, they
Alright.
could still technically unmute themselves and yell stuff.
Right. Hopefully that does not happen. Okay. So to go back to it, Yeah, prediction function is also hard, especially when we consider the regulation of proteins. When we look at the regulatory aspects of protein function, it becomes really poorly understood. So we can predict structure, but what do we need to predict function? We think some of the answer might be in genomic context. So evolutionary processes constrain the sequence structure space for amino acid sequences, and this makes protein structure prediction feasible from a computational standpoint. Evolution also acts on gene content and genome organization. So processes such as horizontal gene transfers, gene duplications, gene losses, inversions, and modular organizations such as operons or co-transcription operons, they all have functional meaning. And we're asking this question of, can we use higher order genomic information to learn a gene's function? So I'm going to introduce this concept of genomic language modeling. This work was recently published and was done in collaboration with Andre, who's actually here today, and Sergey, who's also here, and Elizabeth Kellogg and Pete Gerges. So we took a lot of inspiration from large language models that learn patterns in language without any supervision. So we have a large English language corpus that goes through a large language model such as GPT. And this model, without a dictionary, learns semantics and syntax of natural language. And this sort of foundation model approach can be then fine-tuned to do specific tasks, such as text generation, translation, text to image, image to text, and so on. So we thought, can we teach a model to take in genomic corpus and learn patterns in the genomes to learn the semantics or functions or syntax or regulations of genomic language? And then can we fine-tune that to be able to do certain biological tasks such as novel gene discovery, genetic circuit design, and optimization? So the data set that we used was a metagenomic data set, and we deliberately chose metagenomic data sets because it's the most diverse genomic information that we have access to. And also, importantly, it's the least biased. And I think it's important to sort of address the biases in our data sets because if we think about what kind of data is available on the web, a lot of times it comes from model organisms because that's just how biology research is done. But we need to, in order to be able to build a sort of foundation model or learn biology that applies to general biology beyond just model organisms, we need to be able to teach a model using data that is diverse and less biased. So this particular data set comes from the EBI, European Bioinformatics Institute, and it contains 7 million metagenomic contexts that have up to 30 protein coding genes, and it's sourced from all kinds of environments such as the ocean, soils, and human sewage, and so on. And when we look at the protein coding level diversity of these protein coding genes, there's 31 million proteins that are clustered at 90% amino acid identities. So to train GLM, what we did was we have this 30 gene contig. So these are genes in different colors. And we represent each gene using a PLM embedding. So now, previously these would be different sort of lengths, and now we have a fixed length embedding. And then at time of training, we mask 30% of protein embeddings at random, and GLM is tasked with the objective of reconstructing this protein language model ESM2 embedding given the context. And this is a BERT-like model. And the model is learning the relationships between words by having to reconstruct what the masked word would be. And at the time of inference, what we do is we don't do any masking, and this is a new contig that we don't know much about. And then we represent each protein using PLM embeddings, and then that goes through GLM, and then we can now contextualize protein embeddings. So this is sort of similar to in language models, you can have word-to-vec kind of representation of words, but also you can also have contextualized word that is closer to the meaning of the word, because now you've sort of taken in, oh, this word not only means this in a dictionary, but it also means this in this particular sentence. We can also use attention patterns to be able to parse out regulatory syntax, and I can go a little bit more into that later. Yeah.
Clear. What you're doing, this is at the level of protein sequences, not DNA sequences.
Not yet. Yeah.
It's not like genomic context in terms of DNA. It's where so the context here, when you're saying context, does that mean like where the coding genes are relative to one another?
Yeah, exactly. Yeah. Yeah, so that's this version of GLM, but I'm going to go more into detail about sort of the next iteration and how we're including DNA sequences too. Cool. Yeah, probing function is hard because function is not a one-dimensional thing. And when we first were tasked with this problem of understanding whether this model has learned anything that's biologically relevant, we had to really redesign all the benchmarks to be able to see, oh, this is learning certain aspects of function. So these are all the sort of probings that we did. I'm just going to focus on two that I can continue talking about in the next section of the talk, which is enzyme commission ID prediction, EC prediction, and Operon prediction. So enzyme commission numbers are hierarchical classification scheme, which is very convenient for doing benchmarking. And it's based on chemical reactions that they catalyze. So we're focusing here specifically on the sort of axes of enzyme reaction when we think about function. So what we did was we had protein embeddings that are context-free because at this point, the PLM does not see its neighbors. So these are individually sort of encoded. And then we have GLM embeddings that has taken in all this information from all the context, and now we have a contextualized embedding. And we have, these are sort of, the vector size is the same. And what we're trying to do is to see if the contextualized embeddings carry more or less meaning when it comes to enzyme reaction. And we do that using simple linear probing. So the short answer is GLM representations carry more relevant information for enzyme function prediction. But if we look really deeply into each class of enzyme commission numbers, which there are like 4,000, and there is a huge disparity in the number of proteins in each class. And when we see the actual class distribution, and these are classes where there's a significant, statistically significant difference in the performance of PLM versus GLM. And what we see is that for a majority of these differences, contextualization performs better, but that's not always the case for all of the classes. And there are cases where contextualization performs worse. We need to look into why this is the case. And there may be reasons such as horizontal gene transfer, where the contextualization can obfuscate the information or misannotation too. So yeah, so this sort of gave us an understanding of how much information it can be carried by the same size vector of GLM representation and PLM representations. Yeah.
I guess I should have warned you that we kind of, especially me, interrupt the speaker.
That's okay. Please feel free to.
One thing I'm just really curious about with this result is if you dig into what kinds of enzymes you're getting a lot better performance on the context. Are they sort of these big enzyme machines where they transfer the substrate from one to the next? Or I guess the question more generally, what is it? Is there something to those particular?
Yeah, that's a great question. So yeah, definitely these enzymes that do perform better with contextualization have conserved contexts, and they usually are part of biosynthetic pathways or cascades. So that's one. Another is they have like a specific partner that it always is found next to just by functional signal, I guess. So that's another reason why contextualization may help. Yeah. Cool. So I mentioned attention earlier on. So previous works in large language models have shown that syntactic information can be probed in attention patterns. So in this example, in this language model, BERT model, I think, they were able to reconstruct syntactic subtrees of words just based on attention scores alone. So this is a sort of a zero-shot way of reconstructing regulatory information or word syntax tree information. And we thought, okay, what is sort of a parallel in a microbial genome or the data base that we're currently looking at? We thought, can we look at attention and see if it correlates to operons? So operon is a set of co-regulated genes that are regulated literally through co-transcription. So in this case, gene A, B, C are co-transcribed in a single mRNA. And oftentimes they're involved in a singular task, or they form a complex of a macro protein structure, or they carry out a sequence of reactions where the outcome of reaction that protein A performs is the input for protein B, for instance. So this is what our attention looks like for a random sequence that we found in our data set. And so we have 19 layers and 10 heads. And we looked for if there is any specialization of attention heads that correlate to operon structure. So I forgot to mention that in LLMs, there's been some studies that look at attentions and heads, and they found that certain heads correspond to certain aspects of syntax. So like one head might be specializing in pronouns, or another head might be specializing in, I don't know, adjectives or something. So we looked for attention heads that might correlate directly with operon structure. And we found a couple heads that seem to correlate better than others. And a lot of these heads are found in the shallower layers of the transformer. And when we use equalize operons to see if they see if we can use attention to predict attentions to predict operons, we're able to show that we can predict operons with a pretty good MAP of 77%. So the key takeaway from this first go at GLM is that first, training a GLM is feasible. The initial compression step with PLM overcomes the previous problem of context length. So previously, transformers, if we're trying to just do DNA-based model for genomic modeling, we're not gonna even be able to cover one gene. But now, because we're doing compression with PLM, we can actually cover sort of context length of 15 to 30 genes. So that's pretty exciting, and that gets us to understanding protein-protein interactions and functional interactions between and across proteins. Another key takeaway is that genomic context carries important information for biological function. This is something that we knew as biologists for a long time, but it's good to be able to quantify this and show that contextualization can be represented in vector space and improve downstream functional tasks, function prediction tasks. And GLM captures interaction information between proteins and genes. This is important because molecular interaction form the basis of biological function. So if we're trying to get at the function, we really need to consider the protein-protein interaction or gene-to-gene interaction or gene-to-non-gene interactions and so on. So the main takeaway is that this was a really promising step towards building a context-aware gene-to-function model. And now I got really excited about this project in my last chapter of my PhD. I really wanted to spend the time after my PhD to develop this further and improve it and see if we can really make a difference in annotating a large fraction of this dark matter or genomic dark matter, as we call it. So the work that I'm going to present here is done by our team at Tata Bio, which is a new research nonprofit that is building genomic intelligence to accelerate biology research. So we're dedicated to creating open source AI models, data sets, benchmarks, tools to be able to interpret genomes, not just from model organisms perspective, but across the tree of life, so that we can utilize as much of this information as possible to advance biological technologies beyond just therapeutics. So the three ways in which we are currently improving GLM that we have, I can talk about and we have data for, is first multimodality. So we're including intergenic DNA sequences that you asked about. So I'm going to talk a little bit about that. And then I'm going to talk about training data balancing. And then I'm also going to talk about how we can actually use GLM embeddings well for biology research. Yeah.
Before you get into this, hopefully I'm not anticipating, but just so I understand what you were doing previously with the ESM2 embeddings. So to represent a protein, were you taking the average ESM2 embeddings for each amino acid to represent the protein? So you're just taking each and take the average. And then the second question I have, which hopefully I'm not anticipating, sorry, man, is when you talk about operons, it definitely makes me think of bacteria, and then it also makes me think about how much that database is probably biased toward bacterial genomes versus eukaryotic genomes. And I wonder, in terms of the representation, like, are you learning how bacteria organize their proteins, and does that actually inform how eukaryotic organisms organize the protein? Is it transferable?
That's a great question, and I'm sort of going to touch upon that, but not really. So I can talk about it now. Because our data set is mostly metagenomic, it is focused on understanding protein functions in microbial genomes. But because life is all related, there's homologs. So our sort of thesis is that if we understand a lot about microbial genome, where really most of molecular diversity is found in, then we can actually transfer that knowledge pretty easily at the sort of function level to eukaryotic genomes. We are not modeling eukaryotic genomes right now because of data just not being as prevalent or as balanced as metagenomic data. But one of the sort of benchmarks that we're going to be doing in the future is to see how much functional information we can transfer from bacterial homologs to our eukaryotic homologs. Yeah. Hi.
You know, when this transfer this model, you take like a gain of known function, you put it in like some random context, and you do that millions of times. You can do this. Like, what does the model tell you? You do something.
Could you repeat the question? Sorry.
Like, you have a gain of known function.
Yeah.
And you put it in some context. What history models?
Oh, I see. Okay.
Because you could imagine in metagenomic data, because of sequencing, you get some gene that is known in totally random context.
Yeah, so it depends on the gene, right? So there's going to be some genes that are highly conserved in its context. And there's going to be some genes that are going to be transposons or something, and it's going to be found across all sorts of different contexts, and they're fine doing their thing. So one of the analysis that we did for our first paper was to look at the variance of, given a single gene or sort of gene that's found across multiple times of multiple contexts, look at how varied that context is. And then you can use that information to predict horizontal gene transfer likelihoods. So yeah, so I think, so to answer your question, we can probably look at the embedding, how the contextualized embeddings change across different contexts to see how important the context is for that genome, given the distribution of the data. Yeah.
identify. Is there closer to like bubble or like
So we did not do any filtering for the first step. Yeah, so we were just hoping that the model will be able to identify, okay, if there's like a giant, like a intergenic region or things that are cutting through protein sequences, it should be able to sort of generalize across these sort of noise. It's the same thing as like when you're training LLMs on text from web, there's going to be a lot of junk, but the model sort of learns to sort of filter it out by its on its own. I think so there's going to be some eukaryotic genomes in there, but oftentimes what happens is gene calling is going to be pretty bad. So it's going to look like pretty different or out of distribution from the rest of the data. Yeah. Okay. So I can now talk a little bit about GLM2, which is the version 2 of genomic language modeling. So when we have a metagenomic sequence, what we have is coding sequences interspersed by intergenic sequences. We call this CDS and IGS. And previously, we had this part of the model. And now what we can have is an IGS encoder that separately deals with this modality of DNA. And then, once the metagenomic sequence is sort of parsed into these two modalities and encoded into same size vectors, we can then put them back, interleave them back into that right order and process them through the genomic encoder. And then we do the same masking strategy and then L2 loss on the CDS protein representations. So one way in which we wanted to benchmark what the IGS is doing is to see whether the inclusion of IGS modality improves Operon prediction. What we see here is, the baseline was ESM. ESM doesn't see its context, so we're not surprised that it doesn't do very well in Operon prediction. But once you contextualize it, we see that jump that I talked about in the previous paper. And when we add IGS, we get this huge jump that probably gives a signal of the presence of promoters or intergenic distances between the genes that's giving this extra signal for co-transcription, which was pretty cool to be able to see directly. So what's really cool is, so given this sort of metagenomic context where about half of the genes have unknown functions, when we can predict operons, we can actually now start annotating or transferring the annotations from known to unknown fractions of the proteins. And this is a really high throughput way of and sort of a robust way of predicting or generating hypotheses for proteins that have unknown functions. So these stuffs are, in this example, for instance, these stuffs are domains of unknown functions, are probably associated with methanofuran synthesis, which is a molecule that is part of the biosynthetic pathway. And then we also see a transporter here, which we think is a Deciphering transporter because it's really right next to this TOMB-dependent transporter and lammodulin, which happens to be a rare earth binding siderophore. So when this paper, so now I'm going to shift gears to sequence data sets and our data set. So we expanded our data set to include IMG or JGI's data set, which sort of doubled or tripled our raw data, which we were really excited about. But when we actually look at the data, we see how unbalanced it is. And this paper came out not so long ago that points out this problem that the sequence sampling in the for protein language models is highly unequal, and this has consequences when we use these models for design purposes or for functional predictions. So in our example, this is like 0.1% or less of our data. And when we look at the domain information, a large majority of our context or our examples are from bacteria. But when we have metagenomic data, it's really difficult to align every single alignment is actually really costly computationally, and you can't do all by all alignment. So it's really quite difficult to do data set balancing from the pre-training perspective. You can do that in fine tuning, but if you're trying to build a model that is generalizable across biology, you should really start with a data set that is more balanced taxonomically or phylogenetically. So we noticed that GLM embeddings cluster with the scaffold or genomic sequence taxonomy. So what we're now doing is we're balancing our training data by using GLM embeddings and then sampling relative to the cluster size. And we already see a huge, not a huge, 7% increase in EC predictive performance for a small model, which we hope is going to scale to a larger model. And I want to sort of talk about the use case for GLM. So this paper also came out like a day or two ago, which is sort of alignment-free search and a protein language model embedding based search, which should be faster and should retrieve sequences that are farther away in sequence space or that are remote in homology. So we thought, we're sort of doing the same thing, and we're now calling it GLM search just to sort of draw a parallel here. So can we retrieve sequences, not only using sequence and structure, but also using context similarity? So in this example, our query, this is a UMAP of 5,000 closest embeddings in our small data set. Our query was this PETase, which is a plastic degrading enzyme from a bacterial genome. And so it's here. And this is a UMAP of 5,000 closest neighbors in GLM space. All the gray dots here are things that are found using PLM search. So just using PLM, so not contextualized embedding. So things that if you just looked for 5,000 closest things in ESM space, these are the things that you would find. But then if you start looking for things that are close also in context space, you see these novel sort of clusters appear. And these are things that are probably also functionally similar and structurally similar when we fold it too, but have very little sequence identity. So we cannot even align it to the original sequence. And also really cool is that this blue dot, the coloring scheme is not great, but this blue dot is an archaeal homolog of this PETase. So this would not have been found if you're just doing sequence or structure or PLM search in this case. So we're really excited about utilizing this tool and building a sort of a software layer on top of it so that people can discover functional associations that go beyond sequence and structure, and so that we can start annotating and functionally characterizing the dark proteome. So the key takeaways from this sort of initial improvement GLM2 version 2 stage of our project is that information in intergenic sequences can be leveraged and are meaningful. And they definitely contain different kinds of information that should not be just ignored because it doesn't code for protein. Data balancing is critical for building a foundation model. Because we need to learn generalizable patterns across the tree of life, especially for novel organisms. We're excited about GLM search as a scalable method for propagating and retrieving information, functional information across the proteome. And we're really excited about work to be done here in making this tool usable, but better and scaling this tool further. So I would like to sort of end with a remark that biology takes place in a system, and organismic context is key to modeling and designing biological function. It's really easy to sort of forget about this bigger context when we look at all these cool molecules and folding, but it's in a genome and it's in a cell, and it's important to consider this context when we're designing a new function so that it functions in a environment or a cell. we're really excited about utilizing AI and building these models in open space to accelerate discovery and design of novel biological solutions that go beyond therapeutics, especially excited about sustainability applications. So yeah, with that, I would like to thank you all for listening and happy to take any questions. Thank you.
Yeah, I guess I'll ask a question. I'm kind of curious about the intergenic regions,
Mhm.
and the fact that they help with operons. Do you know if it's like in the between the genes or maybe upstream or downstream of the gene? So I would assume there might be some signal upstream, downstream.
Yeah, so we haven't probed into that level yet. This is sort of like fresh data. But I would imagine it's a combination of sequence signal of like promoters and possibly terminators. But also I think the intergenic distances also that carries weight too. Yeah. So like the size of the actual sequences too. Yeah.
I think I missed this, but which model are you using for the intergenic regions there?
No, we're not using an existing model. We're just using a transformer. Yeah.
Thank you.
Yeah.
What's your model deal with overlapping?
Yeah, that's a great question. So the way we encode this information. So for instance, if there's a gene here, like, so there's an overlap here and there's a second sort of open reading frame here, then it would, the model would see first gene, second gene, third gene, and so on. With the relative directions. So it doesn't really know that it's an overlapping gene, but what we're hoping is that by seeing a lot of data, it's going to be able to sort of generalize to understand that if there's no intergenic regions and if there's significant sort of overlap in sequence, these patterns should be picked up by the model in the end. Yeah, so that's a great question. But that was a design choice that we made to sort of make this easier. Yeah.
Also predict properties about the intergenic regions, like is this a familiar?
Yeah. It's a great question. So we're building out the benchmarks for that. Yeah, it's there's not enough benchmarks for bacterial promoters, unfortunately. But that'll be a really cool to build tool to build because that would have huge implications for synthetic biology. Yeah. Yeah.
Chance to decrypt the genomic search is it referencing mean embeddings?
Mean embeddings?
Like, embeddings.
What do you mean by referencing? For this part, you mean? So do you mean is this the gray parts? So the PLM search, is it done by ESM or?
Like,
Right, yeah. Yeah. So PLM search that we are baselining against is mean ESM to embeddings. Yeah.
So when the GLM, does the GLM search find everything that the PLM search finds, but it finds more? Or are there gaps, just like you showed previously with like GLM 1.0?
Yeah.
Sometimes ESM2 does better.
Yeah, that's a great question. So PLM search seems, so we like did a sort of curve of like retrieval in terms of sequence search. It's pretty much the same, except PLM search gets like less diverse things for some reasons. So yeah, there's like the clusters are a lot tighter. So it's possible, like, so what we're thinking is that GLM search because say, for instance, if there is one protein that occurs multiple times one protein that's very similar, PLM search is going to try and grab all of those. But then GLM search, because of the contextualization, there's like more of a jitter to that embedding space. And then that increases diversification. Yeah, so things are pretty much the same, except there's more duplicates in PLM. Yeah.
I'm not familiar with the PLM search. So when you say the query is, it takes, like, what is the input? What is the output? Like, how do you see this being integrated in like the de novo design, like file?
Yes. So the input is, so we grabbed a sequence from UniProt for PETAs, and then we encoded it using GLM and PLM, and then we get a fixed size vector, and then we're searching it using nearest neighbor search against the embedding database that we already outputted previously. So yeah, so if you like in the lab, for instance, if you're looking for a way to either find a protein that has a different sequence but similar function, then you can do the search to get the original sequences too. Yeah.
You're not getting that query against a database, right? There's neighbors or else.
So the question is, am I doing cosine similarity for the finding the nearest neighbors? Yeah. Yeah, it's cosine similarity, so it's not L2 distance. It seems that cosine similarity does better for retrieving the high sequence matches, yeah, identity matches.
Right? There, there's a large range of presence players. Similar.
Oh, so is there like a threshold drop-off point? Yeah, so this is something that we're still doing in terms of like besides, like user can use like, okay, I want like 10 neighbors versus 1,000 neighbors. But we have to still calibrate where that drop-off is for like homology or its functional homology. But this is something that we still need to calibrate. Yeah, that's a great question though. Yeah. Yeah.
He started going to Fourier transformer. He started picking up, I guess, older interactions for like this
Yeah, that's a great question. This is something that we still need to do work on. So shallower, I meant shallower as in like earlier layers of the thing. And then in theory, what the transformer is doing is learning more complex functional information down deeper. But yeah, that's definitely work we need to do. But in order to do so, we need to design all these benchmarks to be able to see which layer information is the best for what kind of probing. But yeah, I don't have a great answer for like what's in the penultimate layer versus mid layer. But I think from what we are seeing currently, and also with ESM2, the middle layers seem to be really rich in functional information, especially with like EC and also Operon information too. Yeah.
There might be some questions online. You go to.
Yeah. I had one question here, so
Okay.
Yeah.
The results here tend to always be sequence homologs, or do you see, like very random, evolutionarily independent structures that happen to do the same thing?
Yeah, so what we're trying to capture is the latter. There are conversion evolution enzymes that have completely different phylogenies, but seem to have sort of converged in some sort of function and might also have a different structure or structural backbone. And we're able to capture that because the contextual information sort of conserves that information. Yeah. So I don't know how to get the chat. Oh, get chat. Oh, okay. Okay, I think this sort of I answered this part already. Yeah, I think GLM search captures structural information. And we are looking to see if we can sort of benchmark it against fold-seq. But what we're hoping to do is to see if there is something beyond structural information. Also, the problem with structure search, I think, is unless folding becomes really, really fast and really, really cheap, we can't actually fold all our like proteins really quickly. So that maintaining that database alone is harder as opposed to doing embedding based, sequence based and embedding based searches. So that's one advantage that we have for GLM search. Yeah. Yeah, protein-protein interactions. So this is something that we're really excited about. So I guess Operon is just one way in which proteins interact, but we can also detect protein-protein interactions by seeing if there are co-evolving residues across proteins. And this is something that we're currently working on. Yeah, I think that's about it. Oh, there's one more question.
You, repeat the question.
Oh, yeah. Sorry. For the last question. Yeah. That would, so should I do the answers too again or
Oh, no.
oh, okay. Okay.
The next question.
For the next question, how will the GLM model probe into protein-protein interactions? Okay. Yes, so same answer as before, but protein-protein interactions can be captured by looking at co-evolving residues across proteins, not just within proteins. And yeah, and we can benchmark it in silico, but also in vitro too. Yeah. Cool.
Thanks so much. We one more hand. So we still have plenty more drinks and pizza, I believe.